electronic unit. For one, it's affordable, it is eco-friendly, and you'll get more produce per space. Yeah. So for our goals, we wanted to grow herbs using hydroponic systems, research their health and medicinal benefits, develop functional herbal tea blends, create QR codes, infographics, and tea bag packaging. After that, we wanted to compare our antimicrobial results with published research. So for our first project, we made infographics using our own research with reliable sources, and then we got every other team's plants, and we created an infographic with every single team's information. For our plant cultivation, this is where we germinated our plants into rock walls. They would go in the rock wall, then they would be coated with vermiculite to protect the actual seed for germination. After they were transplanted and they were monitored for the pH levels, water levels, and then they were ultimately transplanted into the greenhouse. For our first germination, we had spinach, lavender, sunflower, thyme, and rosemary. Spinach was unfortunately not successful, it died out. And we noticed many mistakes with our first harvest, which we learned from. And we use our mistakes to learn from it. For our second crop, lettuce, mixed romaine, and basil, they were very successful. And here, this is the QR code which you can scan, and you can see the infographics for every single intern's plants. You will see the health benefits and everything with it.
Hello everyone. So I'll be taking you through the development and the production of our tea bags. So I'll show you how we started from drying the teas to grinding the teas to measuring the teas to putting them in their tea bag sachets, and then towards the final packaging of our tea bags. So first of all, we picked out 6 leaves. We picked out basil, sunflower, thyme, lavender, rosemary, and spearmint. So we were able to pick out these leaves and dry them in an incubator at 50 degrees Celsius for 48 hours. And after they were dried for 48 hours at 50 degrees Celsius, we were able to bring them into a hood at room temperature for 70 degrees Celsius at room temperature for an additional 72 hours. And after our leaves were dried, we were able to ground them. So for the grinding process, we put in each herb in the blender. And after we put the herb in the blender, we rinse the blender with 70% ethanol to maintain hygiene. And we also wore gloves in between each herbs that were grinded. And after the herbs were grinded, as you can see on the second picture there, that's the powder form of the grinded leaves. So for the measuring process, for each tea bag sachet, there are 2 grams of one herb. If you see just lavender on the tea box, it has just 2 grams of lavender in the tea bag sachet. And if you see multiple combinations on the tea box, it has 1 gram of each herb inside the tea bag sachet. As for the final packaging, this is how it looks. On the tea box, you see the combination of the herbs on the tea box. It's also on the tag of the tea bag sachet. And then we have our UCNJ logo on it and our QR code, which Meso will be talking more on about.
Okay, so here we have our herbal tea blends and their key benefits. Each combination was chosen because they have complementary benefits. For example, lavender and spearmint help with relaxation and support digestion, while on the other hand, rosemary and thyme help with provide antioxidants and immune support. Overall, all our herbal blends help with different areas such as immunity, digestion, relaxation, and inflammation. We also created QR codes that users can easily scan and access information of each herbal blend. This allows our physical packaging. This connects our physical packaging to the educational information behind it. We created flyers and At the end of this event, if you're interested, you could take one and scan the QR code to learn about our project and the health benefits. Next, my teammate Mia will be taking you through the antimicrobial testing process.
Hi, so we conducted antimicrobial testing to test the plant extracts of their bioactive compounds. So thank you to our mentor, Dr. Big, for giving us the protocol to carry out this procedure. Essentially, we used two different types of bacterial species, 1 gram-negative, gram-positive. For gram-negative, we used E. coli, and for gram-positive, we used Staphylococcus capitis. As you can see in the picture there, at the top, there is a picture of a gram-positive bacterial cell as opposed to a gram-negative bacterial cell wall on the bottom. As you can see in the pink there is a gram-negative bacterial cell which has an extra layer on the outside of lipids, which makes it harder for antiseptics or antibiotics, any drug, to penetrate the cell membrane and kill that particular bacteria. We used 3 different types of controls. For the negative control, we used a sterile disc with 20 microliters of sterile water. We used 20 microliters of each plant extract for the experimental group. And then lastly, for our positive control, we used antibiotic discs, penicillin for S. capitis and ampicillin for E. coli. So you can see here on our TSA plates that unfortunately the experiment did not work. Our results are not conclusive because of the contamination that you can see on the left here. Basically, even though our results were inconclusive, we compared our results to published and existing scientific research which supports the fact that these plants that we did research do have bioactive compounds as seen here that support antibacterial activity. As you can see, there are some bioactive compounds here such as rosmarinic acid, thymol, and most of them are not soluble in water, which is the medium that we use to make the extract, and which is why we got the results that we got. If we were to conduct this experiment again, or if we had had more time, we would have used water and an ethanol-based extract to test the antimicrobial activity of each. Some other things that we could have done is used a millipore filter to extract the possible contamination from the extract itself, lessen the incubation time to prevent possible microbial growth, as well as maximizing our aseptic technique next time.
So I'm going to be talking about the significance and outcomes of our findings. Before I continue, I'm just going to build up on what my teammates said about the antimicrobial results. Most of our antimicrobial findings are a result from already existing published research. Okay. So most of our herbal teas are formulated with antimicrobial, antioxidant, digestive, and wellness benefits. We're able to develop our packaging, and it shows our feasibility in turning our research into consumer-ready herbal tea packaging. We are able to confirm strong antimicrobial potential in most of our herbs, for example, sunflower and spearmint. From some of the results we saw from our research on the internet, we saw that sunflower and spearmint have high antimicrobial properties, and this was what we took into consideration when creating our different mixes for the herbal tea. Okay, we're able to validate traditional knowledge, traditional uses with our lab evidence. We're able to develop functional teas with different benefits such as antimicrobial, antioxidant, digestive, and calming benefits. That's why we created the QR codes you see on our tea boxes. When the consumer takes the packaging and you scan the QR code, you're going to see the basic different benefits of whatever mix you basically pick. For example, you have a lavender and rosemary mix, you're going to be seeing maybe the psychological support, helping with mental, any mental stress, and different things like that. We're able to identify high potential herbs for future agribusiness scaling, such as sunflower and spearmint, like I said before. So we have alignment with agribusiness goals. As the biology team, we basically try to align biology with business in a general. For the first one, we have value-added product development. We're able to transform our raw herbs into herbal teas with different wellness benefits, and we're able to support this with the antimicrobial data we found, not just from our results, but also from published existing research. And number two, we have evidence-based market differentiation. What basically differentiates us from generic herbal tea blends are basically that we use antimicrobial results to back up whatever mix we have in our products. Number three, we have sustainable local resources. All our herbs are grown from UCNJ greenhouses, UCNJ farms, UCNJ hydroponic towers, and we also were able to reduce supply chain costs and support community growth by purchasing most of our seeds from local farmers in Union County. Commercial scalability, because of our simple extraction methods and our straightforward packaging, we're seeing the opportunity to be able to scale our project into a larger scale in the future. For our acknowledgements, we'd like to express our gratitude to New Jersey Pathways, Fluxbase, Amanda, Dr. Mohammed, and Dr. Smalls, most of our interns at the Agribusiness Internship, UCNJ laboratory staff, and UCNJ as a whole for all your support in everything. Thank you. And at the end of the event, if you would like to taste any of our tea mixes, we have them over here in the kettle, so you can be able to be the first person to experience the consumer experience as